Section 5 is chemical properties of monosaccharides. The first thing we have here is reducing sugars. And these are basically just sugars that can, as the name suggests, reduce other substances. They in turn get oxidized in the process. You might remember the picture here. This is exactly what we saw in the organic chemistry chapter with when we talked about the Benedict solution for the testing for the presence of aldehydes. You can see the aldehyde gets oxidized into a carboxylic acid there. But this aldehyde happens to be a sugar, and so the sugar gets oxidized from an aldose into a carboxylic acid form of, in this case, glucose, which is D-gluconic acid, the product of that. And anyways, this type of reaction, or the ability of subsugars to reduce other substances or to be oxidized, is actually responsible for the Maillard reaction, which is what makes certain things you bake or cook taste really good. It's like the caramelization process of sugars, essentially. So the reason you put barbecue sauce on your grill while you're cooking it is the Maillard reaction. It tastes so much better when you do that, or really just any food as it's cooking. It hits a certain point, I think about 155 degrees Fahrenheit. The Maillard reaction begins, and the chemical nature of these sugars changes. They get oxidized. But not all sugars are reducing sugars. All monosaccharides are reducing sugars. Many disaccharides are, and only a few polysaccharides are. So in some ways, the Benedict solution is a test for if it's not really about there being an aldehyde, like if you know it's a sugar, you can test whether it is a polysaccharide, a disaccharide, or a monosaccharide. 13.6 is on disaccharides, and as the name suggests, there are going to be two saccharides, two monosaccharides to make up a disaccharide. Saccharin essentially comes from the Greek word for sugar, so two sugars here. And the disaccharides form from these two monosaccharides. They react together to make what would be called the glycosidic bond, which I've highlighted in blue there at the bottom. That's the glycosidic bond. Okay. And so you can see the alcohol on carbon 1 has reacted with the alcohol of carbon 4 in this case, and it has emitted a water molecule. So the oxygen and hydrogen there plus that oxygen are going to leave to form the water. The oxygen there is going to basically attack that position. So if I was to make that a red oxygen, this would be red right there. Okay, and so that's, you get this carbon oxygen carbon bond. That group is the glycosidic bond, and the terminology here, alpha 1 to 4, that just tells you the positions that they occurred, because it can be different. Couple other examples of disaccharides. We have galactose combining with glucose to make lactose. Lactose makes lactose with glucose. And what your body does when you drink milk, you get this lactose in there, and your body, if you're not lactose intolerant, you just digest it, you metabolize it, you kick off the galactose. Your body then converts that galactose into glucose, and so you actually end up with two glucose molecules there that then goes through respiration and you convert to CO2. And in fact, really all sugars, no matter what the monomer form of it is, you will break them down, and then they will get first converted to glucose, and then the glucose gets converted into CO2. Here, if we combine glucose and fructose together, we're going to make sucrose. That's table sugar. So table sugar, this stuff you add to everything, is fructose and glucose, a disaccharide of that. And you'll notice that the fructose is a 5-membered ring, not a 6-membered ring right there. And I'm going to emphasize here now that you don't need to worry about the alpha and the beta so much. Like, I'm not going to say, is sucrose alpha D glucose? Is it alpha L glucose, beta D glucose, or beta L glucose? And is it alpha D fructose or beta D fructose, or is it alpha L fructose or beta L fructose? That stuff I don't really care about at all. And same thing with the glycosidic bond. It says here alpha beta 1,2 glycosidic bond. That's just superfluous information. Section 7 is polysaccharides. So these are things that have lots and lots and lots of monosaccharide units combined together. And the first one here we have is essentially starch. And starch is made up of 2 different types of sugars. Essentially, we have the amylose, and that's about 20% of it. And then there's amylopectin, and that makes up basically the other 80%. And if we look at this, we have an unbranched chain of amylose, so the amylose is unbranched, and then the amylopectin is branched. You can see that the amylose forms a helix, a single helix, not a double helix like DNA. So you get that helix in there. Whereas the amylopectin, also called glycogen, that one has the branches, and so it forms a jumbled mess. There's no helix or beta pleated sheet or anything like that. You get branches in multiple ways. And so, yeah, there might be parts there that are helical, but since it's branched, it doesn't really work perfectly like that. When you ingest starch, your body breaks apart each of the monomeric units of the polymer and converts them into glucose in the acidic solution of your stomach. Okay, and starch is primarily used for energy storage. Glycogen is the next polysaccharide here. This is basically animal starch. The other one, you could see the potatoes in there. That was plant starch. We just call it starch. But glycogen is made up from units that are also made of glucose, like with the plant starch, but the branching is much different. For example, the plant starch has branches every 25. All right. I'm going to say every 25 units, whereas glycogen is every 10 to 15 units. It branches off. So this part right there would be a branch off of the bottom part down here. Okay. And yeah, I wrote in here alpha 1,6 glycosidic linkage, but I'm not going to test you on that at all, so don't worry about it. Glycogen is stored in your liver, and basically as you eat food that has sugar in it, some of the glucose gets converted into this glycogen because your body knows that you're going to need to store energy, and you need to store it more short term sometimes. The lipids are going to be long-term storage of energy. Glycogen is short-term. So basically you eat sugar and you don't immediately use all the energy you get from it. That wouldn't make sense. We're not crazy. So we take that sugar and then we store it as glycogen. When we need glycogen, it gets released from our liver and our body breaks that down into glucose, and then we can use those pieces for energy. Okay, and this is actually what happens when you wake up. Your body releases a whole bunch of glycogen so that you have energy to wake up. The last polysaccharide is cellulose, and cellulose differs from the other two in that it is unbranched, and even if it was amylose from the plant starch, which forms the helix, the cellulose groups do not form a helix. They stay flat, and the groups of them, like in multiple different chains, they're going to be parallel to each other, which means they can stack very easily. And so cellulose is the major structural component for wood and plants, like the stem of plants and leaves and all that stuff. Paper, for example, is almost entirely cellulose. Cotton is essentially cellulose as well. So that is it for chapter 13, carbohydrates. We covered everything here.